Alrighty, in this video I'm going to talk about experimental design and endosymbiotic theory. So I'm putting two things together. First up is experimental design. So I want to remind you of three key things that you are guaranteed 100% to be asked about on the AP exam. Independent variable, dependent variable, and control. So let's kind of slow down and talk about each one. The independent variable is the thing or the condition that the scientist is changing in the experiment. So the scientist maybe is using different amounts of light for different plants or different types of plants or manipulating something about an experiment, and that is the independent variable. The dependent variable is the thing that the scientist is measuring. So if we're looking at plants, maybe we're measuring like the amount of carbon dioxide that they're using or the amount of oxygen that they're releasing, or maybe the growth, how tall they are over time, or the number of leaves that they have. Whatever we're measuring, that's our dependent variable. The control is whatever does not get the treatment. In other words, it's something to compare to. So like if I'm looking at how the amount of light affects how a plant grows, maybe my control is a situation where there's no light. Or if I'm looking at how different fertilizers affect a plant growth, maybe my control is without fertilizer. So it's something so I can compare to a baseline to see how the thing I'm changing is even affecting what I'm measuring. An example is from the mock exam, where they gave you a graph where we had SEPin1 concentration on the x-axis and percent of cells viable on the y-axis. A trick: the x-axis is always going to be the independent variable, the y-axis is always going to be the dependent variable. Also, the SEPin1 concentration was the thing the scientist was changing, and the percent of cells viable was the thing that the scientists were measuring, like how many of the cells were living. The control was the lowest amount of SEPin1 concentration that they used. So I think in that case it was like 0.1. Also a good control would be no SEPin1, like how many cells survive when there's no SEPin1. That would be an excellent control. A lot of you got confused with what we previously called controlled variables, which a better word for is constants. But I think that on the exam, if they're asking you, or I know that on the exam, if they're asking you for control, they mean this. They mean the baseline, the thing that you're going to compare to. If they're asking you about a variable or something you could keep constant, they're going to use that language instead. So another example is researchers want to see, this is a totally made up experiment, but researchers want to see how caffeine, independent variable, affects people's growth, dependent variable. So how something affects something else. They have 3 different groups with people drinking different amounts of caffeine. So different amounts of caffeine, that's our independent variable again. And they measure their height over 20 years. So again, that's the dependent variable. They're measuring the height. So question for you, what would be a good control for this experiment? I'm going to pause for a second so you can think about it. So maybe like having a group of people where you're just giving them water, or they're not drinking any caffeine, or they're drinking like decaffeinated coffee or something like that. So you want a baseline to compare to, right? So that would be an example of a control. Another thing I want to point out in terms of experimental design, more data equals more reliable and better results. So for example, if we're doing this experiment, we would rather test 100,000 people than just 10 people. So the more data that you're gathering, the more valid and the more like believable the results you're seeing are. Because if you just do a couple people, who knows, like maybe like the people that you randomly assigned lots of caffeine are from a family that grows really tall anyway. So you want to have a lot of people and a lot of data points to make your data more reliable and valid. So next up, changing gears entirely. Endosymbiotic theory. So as a reminder, this is when one prokaryote engulfed another prokaryote and we ended up with a eukaryote. So with a membrane-bound organelle with one prokaryote living inside another one. So initially, they were just living like symbiotically. They were helping each other out, but that turned into a eukaryote. But that's where the symbiotic part of the word comes from. Endo comes from, this is endocytosis. This prokaryote is endocytosing this other prokaryote. So I want to point out that this is a double membrane here. So part of the evidence that we have that endosymbiotic theory happened is that we have organelles such as chloroplasts and mitochondria that have a double membrane. And also within chloroplasts and within mitochondria, they have their own ribosomes and their own DNA. So that's further evidence that they used to exist on their own. Another kind of set of reminders is the difference between prokaryotes and eukaryotes. So prokaryotes were the first form of life, whereas eukaryotes are newer, right? Because prokaryotes came first and it took prokaryotes to make eukaryotes. Prokaryotes have no membrane-bound organelles. So for example, no nucleus, no endoplasmic reticulum, no Golgi body, no lysosomes. They do have ribosomes because ribosomes are not a membrane-bound organelle. Whereas eukaryotes have membrane-bound organelles like nucleus, endoplasmic reticulum, etc. Prokaryotes have free-floating DNA because they don't have a nucleus, and their DNA is typically circular. Whereas in eukaryotes, the DNA is in the nucleus and it is linear. Another kind of side note is prokaryotes, because they don't have all of this nonsense of the organelles and such, they divide faster. Whereas eukaryotes divide slower. So this is a benefit for eukaryotes because sometimes it means that their DNA replication and such is a little bit more, has a little bit higher fidelity, like it's better, whereas prokaryotes are kind of dividing faster. All right, and that is all for this video.